mean? It means phenylalanine. Okay?
Okay.
Let me say it now in English. Ooh means phenylalanine. Class, this is not a spoken language. Some of these words you can pronounce, but you don't pronounce most of them. But U means phenylalanine. That's what that three-letter word means. Let's try another one. How about C? C. That's that one.
Proline.
That means proline. Can you see what I'm doing here? On this chart, you see every possible three-letter word that can be made for four things that can be repeated. Now, mathematicians, four things, taken three at a time, where you can have repeats. How many words can you make? I told you I was going to mess up, because no, that's not the answer. You can make, here's how you do it. How many can fit in the first position? How many different possibilities?
4.
4. How many in the second?
4.
4. And in the third?
4.
4. So you multiply them together. So quickly. 4 times 4 times 4. That is?
64.
64. There are exactly 64 words in this language.
Why do some of them say stop? Stop.
I'm glad you asked, but you're getting ahead of me. First of all, you need to notice that up here, all 20 amino acids are here. Now, you may not have recognized that, but if you'd been in my chemistry class, you would have already memorized all 20 amino acids. And you could name them. And you could tell me their chemical structure. Every amino acid's up there. In fact, there are repeats, aren't there? There are 5 or 6 leucines. So you say, why would you have repeats? Well, it looks to me like a backup system. Huh? There's more than one code for leucine. In addition to that, she noticed something else. There's a code AUG that means start reading.
Okay.
And there are 3 codes up there that tell you to stop reading.
Okay.
Now, I need your careful attention. Everybody's eyes on me. I've got a string of amino acids. What is that?
Protein.
That was the right answer. I'll say it again. I have a string of amino acids. What is that?
Protein.
A protein. So it's amino acid, amino acid, amino acid, right?
Yeah.
That's a protein.
Right.
And protein is recorded, that amino acid sequence is recorded on the DNA strand. The question is, when do you start reading and when do you stop reading? Well, there's a code for that.
Is it like zeros in the
No, it's those particular 3-letter words. You come to a UAG, if it's properly spaced, it means start reading. And then from there you go read, amino acid, amino acid, amino acid, amino acid. Where do you stop? When you get to one of those 3, either UAG or what?
UAA or UGA.
UAA or UGA. Any of those 3 will tell you to stop reading. So you stop reading here, you started reading there. In between, you have a string of amino acids, which is the code for a protein. Exactly right. And that's how, class, in a living cell, we know how to build proteins. Because they're coded in the DNA. Are you with me? This is revolutionary. This came to light in the late 50s and early 60s and on into the 60s. And people were blown away by this. But did they come to the conclusion this had to be designed? Now, a lot of folks say, no, isn't it interesting what nature did? I'm sorry, I don't have that much faith. Not nature. All right, so what have you got here? You have a language with 4 letters, 3-letter words, and those 3-letter words in a sequence tell us a code for
Protein.
Exactly right. So here's my question, class. Ladies and gentlemen of the jury. What if a member of SETI looking in a radio telescope, found from outer space a message containing the genetic code, and they learned how to decode it and read it, what do you think they would say?
Isn't that amazing?
There is intelligence.
Yeah, they would say somebody out there is intelligent doing that. Nature doesn't do this. But when it's in every cell of every living thing, we say, isn't it interesting how that evolved by natural causes? No, I don't think so.
Missing the forest for
The SETI organization and any other reasonable people would certainly conclude something like the genetic code came from an intelligent source. So why don't we think cells come from an intelligent source? We should. But ladies and gentlemen, all we've talked about right now is just that language and how it builds three-letter words which ultimately show you the code for building proteins. That is hardly any of the story. That's just a veritable tip of the iceberg. So what I'm going to do for you in the next few minutes, I have a little video clip you're going to come help me with that shows, and this video clip, by the way, comes from the website for this book. You can go look it up and listen to it again. And there's a guy moderating it, but I'm going to moderate it here. But you can go listen to him and look at it again. So here's the video clip from the Signature in the Cell website. So look, there's the DNA molecule. You see it? I wish I had a bigger pointer, something brighter. We'll do the best we can. There's the DNA. And you see the cross strands? All right? Let's go ahead. So this is a cartoon, you understand. Yep, get rid of the, yeah, thank you. So can you see the C, A, G, and T in here? And the cross pieces?
Yep.
Okay. And so it's just showing you the DNA molecule. And then watch, stop right there. You see this glump here? That glob of stuff? That's a glob of, what do you think? Yes! And do you see the different colors, green and yellow and brownish? That's 3 different proteins that ride along the DNA. Okay, one of them is called RNA polymerase. If it's a polymerase, it's an enzyme. And you learned earlier that enzymes are, thank you, that was not loud enough. You learned that enzymes are proteins. Correct. So what you've got here, class, is you've got 3 proteins in it. There's really more than 3, riding along the DNA strand, okay? If you don't have those proteins riding along the DNA strand, you ain't building anything. Pardon my English. Okay? Next, go ahead. Okay, now watch what happens here. Here comes another one. Do you see how that wrapped around there? And see the one with the little fly left there? Stop, right here. Now, this is more what it looks like. Those other things were cartoons. This kind of globish thing is more what it looks like. That's a bunch of proteins clinging to the DNA, each of which has a very specific purpose. Purpose leads to design. All right, so they're riding along the DNA molecule. Go ahead. So here's what happens. Stop right there. Can you see that the DNA is now unzipped? Yeah. And it's exposing the 2 sides. You know, class, what it takes to unzip DNA? Thank you very much. Can I ask you again? What does it take to unzip DNA? Proteins. That RNA polymerase rides along it, and it literally allows it to unzip. Now, I'm jumping way ahead here. When you get through using the DNA that's unzipped, guess what it has to do? It's got to zip back together. Guess what it takes to zip it back together? Proteins. A different protein. You ever have trouble zipping up a zipper? It's embarrassing for men especially. Sometimes you have trouble zipping things back up. Did you know that can happen with this stuff too? Because it's very complicated. So it takes a protein to get it zipped back together properly. All right? Kind of in your mind, keep a kind of a total running of how many times you've answered the name protein. Okay? All right, here we go. So now here's what's happening. The side is exposed, and do you see we're coming up and we're setting a little copy Stop right there. This copy is copying the information that's here. Everywhere there's an A, what do you think's over here? A G. And if there's a T over here, there's a C over there. So it is the exact mirror image of the information that's on the DNA. And what you're doing is you're building a copy of that information, and that is called messenger RNA. Got it? When that RNA gets finished making that copy, it's got the exact mirror image of that string into the RNA. Totally amazing.
Wait.
Yes, ma'am.
So what?
See this RNA right here?
Yeah.
One piece at a time, it's building the mirror image of that information, and then it's connecting up by chemical bonds.
So it becomes part of the new DNA. It forms a new.
It's an RNA.
It's an RNA, and then the RNA goes away.
You're going to see in just a second. Getting ahead of me.
Okay.
But that's a good question. Yeah. First of all, you got to copy the information. That's called transcription. Right? The information is useless unless you can copy it and do something with it. This is called transcription. So you're copying the information onto an RNA molecule. And it's the exact mirror image of what's on the DNA. Okay? Let's go on. Now watch closely. Oh, pause just a minute. Here's another problem with this. I hate having to look at me. The problem with this cartoon is it's way too slow. That stuff that you're watching right there as the RNA builds, you know how fast that happens? Watch. That was way slower than it happens. That ought to be so fast you can hardly see it. That's what happens. The copying of this thing goes, zoom, and it's done. And then it does the next one, and the next 10,000. How do you think your cell builds 80% of the proteins in it in a typical bacteria in 10 minutes? It's incredible. So just understand, this is like a slow train to China. It's not the way it works. It's zoom. Okay, go ahead. So here's the RNA. Stop right there. Notice here's the RNA strand. Now it's being released out into the cytoplasm of the nucleus. After it's made its copy, we call it messenger RNA. Good name, isn't it? It's taking the information from the DNA and taking it somewhere. So the transcription is now finished. It took, class, for the transcription about 15
Proteins.
Thank you. Y'all are pulling back on me. To build the copy, the transcription, takes about 15 proteins. One of them's RNA polymerase. Incredible. But that's not even gotten it anywhere except the copy made. Now it's got to migrate. So the RNA, right there, migrates. And let's watch it. Here we go, migrating through the cell cytoplasm. But the problem is, stop right there, there is a cell membrane around every nucleus in any respectable cell. How do you think messenger RNA gets outside of that membrane? Well, they've got holes.
Proteins.
Yeah, you're getting ahead of me. Guess who guards the hole?
Proteins.
That's exactly right.
I'm good at this.
Yeah. Here comes the RNA up. Now, do you think any respectable cell is just going to let anybody go through those holes? No. Only who's allowed. And guess who guards the gate?
Proteins.
Watch. So you come up to this hole right here, or wherever it is, go ahead. Watch the RNA. Here he comes. And he says to this hole right here, I need through. Watch this protein. Oh, come on in, dears. You may pass through until you are finished. Stop right there. And when that RNA gets finished, the protein shuts back up again. And class, on the other side of the hole, this is the cell nuclear membrane. On the other side, there's also a hole, right? What do you think guards that?
Proteins.
Absolutely, on both sides. You don't just let anything going out of the nuclear membrane. It requires protein. All right, now here goes the RNA. And he comes out into the cytoplasm of the cell. Go ahead. And then watch, as he comes out, he's picked up by, watch, something just closes in on it. See this? There you go. That's, stop right there. I didn't make you stop saying that. That thing that closed in is called a ribosome. Okay? And the ribosome looks kind of like two ducks hugging each other. But it's really closing in because it wants to take the information and read it. So you've had it transcribed, you've had it migrate, and now you're having it translated. Okay? Down inside. And now we're down inside the ribosome, and you see this is the RNA, and the code is CAC, AGA, CUA, CCG, ACC. Each one of those three letters is an amino acid, right? And the string builds a
Protein.
Exactly. Now, how do you do it? This is just the code. Watch, class. This guy right here is called a transfer RNA. Have I said anything about transfer RNAs yet? No, that's another 6 lectures. And I'm not talking about that. But you got to have them. The transfer RNA picks up the specific amino acid out of the cytoplasm, but you better be eating or you won't have it. And he brings it in, and guess which amino acid he picks up here? It's the one that exactly agrees with that code. Now I lost you. So I'm going to say that again. The transfer RNA picks up an amino acid in the cytoplasm because you ate it, and it brings it inside the ribosome, but it brings the exact one needed for that 3-letter code right there.